Hello students, welcome to SK Biology. This video is about DNA fingerprinting, a very important technique in molecular biology and forensic science. Before starting, look at your finger for a moment and press your thumb on ink and then stamp it on a paper. You will see unique lines as shown here in this figure. This is fingerprint, and you might be aware that no two people in the world have same fingerprint, not even a siblings. Fingerprints are unique to each individual. In the same way, our body also have something more powerful biological fingerprint in the DNA of each person, called as DNA fingerprint. Let us understand it in detail. DNA fingerprinting is a molecular technique used to identify individuals on the basis of unique DNA pattern. As that of normal DNA fingerprint, no two individuals will have same DNA pattern. The DNA pattern will be unique for each and every person. Let us understand it with example. Suppose there are two individuals A and B, and we want to check their DNA pattern. Firstly, we need to isolate DNA from each person, and then this DNA is fragmented using molecular scissor called as restriction enzymes. After cutting, each person will produce different pattern on agar gel. This pattern is due to differences in DNA sequence, the basis of DNA fingerprinting. These unique patterns are used for individual identification. Now, the question is, how does these unique patterns formed? Answer is, special repetitive DNA sequences present in each person. Mainly, two types of repetitive DNA sequences are responsible for this pattern formation. One is VNTR, which is variable number of tandem repeats, and second is STRs, that is short tandem repeats, which we have studied in case of satellite DNA also. These both repetitive sequences are highly polymorphic, meaning they vary from person to person just like our thumb impression. Here in DNA fingerprinting, the basis of variation is because of polymorphism in DNA sequences. I repeat, the basis of variation is because of polymorphism in DNA sequences. This technique was discovered by Alex Jeffrey in 1984, and this discovery completely changed forensic science. Now, let us understand how DNA fingerprinting can be done, that is, working of DNA fingerprinting. The first step is collection of sample. Sample can be anything containing DNA, like blood, hair, semen, saliva, etc. After collecting sample, the second step is isolation of DNA from sample in pure form. After isolation, the third step is amplification of DNA by PCR, that is polymerase chain reaction discovered by scientist Karen Mullis. This technique generates copies of DNA outside cell. After amplification, the DNA is fragmented in next step with the help of restriction enzymes, and then they are separated on agar gel to get DNA pattern. The basis of unique DNA pattern is restriction fragment length polymorphism, also called as RFLP. Once the unique DNA pattern is available, this DNA fingerprinting pattern can be used for various functions or applications, like in forensic science for solving crimes, for paternity testing, for analysis of genetic disorders. DNA fingerprinting can be also used for wildlife conservation and also for victim identification.